Our abstract basically takes advantage of the fact that as an institution we have collected a large, many hundred patient cohort of patients with metastatic breast cancer. Most of them have hormone receptor positive metastatic breast cancer, but there's also some triple negative breast cancer there. And so we were interested within that large cohort of identifying patients who have a mutation in either BRCA1 or BRCA2, so the main breast cancer susceptibility genes that we all think of, and were exposed to treatment with either a PARP inhibitor or a platinum type of chemotherapy, which are types of medicines that are sort of particularly targeted for effectiveness in a BRCA1 or a BRCA2 mutant patient. And so we picked out 8 of those patients from the large cohort of patients with metastatic breast cancer, and we did sequencing on tumor samples that were available both after the patient had acquired resistance to the PARP inhibitor, the platinum therapy, and some time point before that, at the time when the patient was initially sensitive to the PARP inhibitor or the platinum therapy, as a way of basically trying to look in the genome for reasons that these patients who initially responded to these sort of targeted therapies would have initially responded and then subsequently become resistant. It's a small cohort again, it's 8 patients, so there are certainly findings that need to be looked at in larger cohorts. But what we found is that in 4 of 8 patients, so 50% of the patients, it looked like the patients acquired resistance to the therapy because they basically, their cancer cells found a way to become no longer BRCA mutant. So they developed what's called a reversion, meaning that they acquired a secondary alteration in BRCA1 or BRCA2 genes, such that instead of being BRCA deficient in the way that they had been at the beginning when they were responding to the therapy, they ended up being BRCA intact, and therefore they weren't susceptible to the therapy anymore, and so they progressed and had to move on to a different treatment. So that's what we found in 4 of the 8 patients. And then in 2 of the 8 patients, we found evidence in the sequencing results that they may have acquired resistance to the PARP inhibitor or the platinum by increasing a part of DNA repair processes called end resection. And so that finding was interesting and potentially important to us because it maybe points to another mechanism, another aspect of biology that you could target in combination with the PARP inhibitor, the platinum, in order to prevent or overcome resistance from developing to the original therapy. Because of course what we ultimately care about is not just figuring things out in the DNA sequence of tumors, but actually being able to use that to figure out why therapies stop working and then make them work better for our patients. Currently in the clinic, I can speak mostly to breast cancer because I'm a breast cancer doctor. In breast cancer, our use of PARP inhibitors and platinums in patients who are BRCA1 or 2 mutant is relatively recent. So the first two PARP inhibitor drugs were actually both FDA approved in breast cancer in 2018. So in breast cancer, I would say that we haven't had a lot of insights, at least not breast cancer specific insights, into why resistance develops and how to manage it. So honestly, we sort of manage those patients in the way that we manage other metastatic breast cancer patients, which is that based on their disease status and their clinical status at the time of disease progression, you decide from the menu of options what treatment makes sense to use next. But I think up to this point, we've been lacking ideas about exactly how to think about why they became resistant so that we could be rational in what treatments we use next, which is definitely still a work in progress, but that's what we're working towards. Another thing that was exciting in our results, and another thing that we looked into, again, these are preliminary results, it's a cohort of 8 patients, but we looked at the use of the biomarker called RAD51, and so RAD51 is a protein that's involved in DNA damage repair by homologous recombination, so it's basically in the same pathway as BRCA1 and BRCA2. And what we found, and what others have found as well, and we corroborate, is that you can use the presence or the absence of RAD51 as a marker of whether BRCA1 or 2 is working or is not working in a given tumor, and whether the DNA damage repair pathway of homologous recombination is working or is not working in a given tumor. And The exciting thing that we found is that you can determine that by doing a simple stain for RAD51. So, I'm describing results from sequencing tumors. That takes months. Then you have to analyze it. That's obviously not going to be deployable in real time, at least not in 2019 in the clinic. But something like RAD51 staining, where you just have one slide, you do a simple brown stain, and you look for the presence or absence of a marker, that's something that we could really deploy in the clinic. And our results suggested that maybe that's a meaningful clinical marker that can predict response or resistance.
Great. And what are the next steps for this research?
So, I think the main thing is that it needs to be looked at in larger cohorts, and it needs to be replicated and taken farther, because I think our results are exciting, and we're hoping that they ultimately translate into benefit for patients and helping these two classes of therapies work better in patients with BRCA1 or 2 mutations. But it's an 8-patient cohort, and certainly, we need larger cohorts of patients who have participated broadly in clinical trials of PARP inhibitors or platinums, cohorts from other institutions, and we need to do sort of similar analyses both of genomics and of RAD51 in those bigger cohorts.